Within every living cell lies a microscopic marvel. This is DNA, the molecule that carries our genetic instructions. It determines our traits, from the color of our eyes to our susceptibility to certain diseases. The story of how we unlocked this blueprint is one of scientific ambition, collaboration, and controversy. Enter James Watson and Francis Crick, two ambitious scientists working at the University of Cambridge. Their relentless pursuit of this mystery would change the course of science forever. This is the story of how the double helix, the elegant structure of DNA, was revealed. By the early 1950s, the race to decipher the structure of DNA was heating up. Among them were Rosalind Franklin and Maurice Wilkins working at King's College London. They were using a powerful technique called X-ray diffraction to study DNA's structure. Meanwhile, in Cambridge, Watson and Crick were building physical models, trying to piece together the puzzle of DNA's shape. The competition was fierce, but the pursuit of knowledge was the driving force. The discovery of the double helix would be a pivotal moment in scientific history. Before Watson and Crick's groundbreaking discovery, scientists had been grappling with the fundamental question of heredity for decades. Early experiments, like those conducted by Frederick Griffith in the 1920s, hinted at DNA's role in inheritance. Further research by Oswald Avery and his colleagues at Rockefeller University in the 1940s provided strong evidence that DNA was indeed the carrier of genetic information. Understanding the structure of DNA would be the key to unlocking the mysteries of heredity itself. As scientists delved further into the study of DNA, they began to unravel its basic components. Each nucleotide consisted of a sugar molecule, deoxyribose, a phosphate group, and one of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. In 1950, Austrian chemist Erwin Chargaff made a crucial observation that would later prove essential to Watson and Crick's breakthrough. Meanwhile, at King's College London, Rosalind Franklin and Maurice Wilkins were employing X-ray diffraction techniques to study the physical structure of DNA. Franklin, a brilliant crystallographer, obtained particularly clear X-ray diffraction images of DNA. The scientific community was captivated by the mystery of DNA. At the Cavendish Laboratory in Cambridge, James Watson, a young American biologist, and Francis Crick, a British physicist, had teamed up to tackle the DNA puzzle. Unlike Franklin and Wilkins, who focused on experimental approaches, Watson and Crick relied heavily on model building and the interpretation of existing data. They were acutely aware of the competition, particularly the work being done by Linus Pauling in the United States. They were determined to be the first to crack the code of life. In the hallowed halls of Cambridge University, a scientific partnership was igniting. James Watson, a brash young American, crossed paths with Francis Crick, a British physicist. Despite their differences, Watson and Crick found themselves drawn together by a shared passion for the mystery of DNA. They spent countless hours in animated discussions, their conversations echoing through the corridors of the Cavendish. The quest for the structure of DNA was not without its stumbles and setbacks. Watson and Crick built model after model, only to have their creations crumble under the weight of new data. One early misstep involved their initial assumption that DNA might adopt a triple helix structure. Undeterred, Watson and Crick pressed on, their determination strengthened by each setback. The path to the double helix took a decisive turn when Watson and Crick gained access to Rosalind Franklin's groundbreaking X-ray diffraction images of DNA. One image in particular, known as Photo 51, revealed a striking X-shaped diffraction pattern that hinted at a helical structure. The clarity and detail of Photo 51 immediately caught Watson's eye. Armed with this new information, Watson and Crick returned to their model building with renewed vigor. Their model, an elegant double helix with 2 intertwined strands, was a revelation. The discovery of the double helix structure of DNA marked a turning point in our understanding of life itself. Published in the journal Nature in April 1953, Watson and Crick's landmark paper ignited a revolution in biology. The double helix provided a clear explanation for how genetic information was stored and replicated. The implications of Watson and Crick's discovery were far-reaching, extending far beyond the confines of the laboratory. The triumph of the double helix model was not without its shadows. The brilliance of Watson and Crick's achievement was built upon the work of Rosalind Franklin. Her expertise in X-ray diffraction produced some of the most detailed images of DNA ever seen. The story of Rosalind Franklin is a poignant reminder that the pursuit of scientific knowledge is also a human endeavor. The unveiling of the double helix structure was met with widespread acclaim. However, the shadow of Rosalind Franklin's unacknowledged contribution loomed over the celebration. The use of Franklin's data without her explicit knowledge or consent ignited a debate about scientific ethics. It highlights the importance of transparency, collaboration, and proper attribution in scientific research. Rosalind Franklin's legacy extends far beyond the controversy surrounding the discovery of the double helix. She made significant contributions to the understanding of coal and virus structures, in addition to her groundbreaking work on DNA. Her meticulous approach to research and unwavering determination continue to inspire generations of scientists. Franklin's contributions to science have been increasingly recognized and celebrated. Numerous books and articles highlight her brilliance and the challenges she faced as a woman in a male-dominated field. Institutes, buildings, and scholarships have been named in her honor, ensuring her story is remembered. Her legacy inspires us to create a scientific community that values diversity and inclusion. By embracing diversity, we pave the way for new discoveries and a deeper understanding of the world around us. The unveiling of the double helix structure of DNA was akin to handing scientists a brand new set of keys to unlock the secrets hidden within the very blueprint of life. With the elegant structure revealed, researchers could finally begin to decipher the language of genetics, understanding how this molecule, with its elegant twists and turns, orchestrated the symphony of life. The double helix, with its two intertwined strands connected by a specific pairing of chemical bases, adenine with thymine, guanine with cytosine, held the answer to a question that had puzzled scientists for generations. How is hereditary information passed down from one generation to the next? The beauty of the double helix structure lay in its simplicity. The specific base pairing provided a mechanism for faithful replication. When a cell divides, the double helix unwinds, each strand serving as a template for the creation of a new complementary strand. The result? Two identical DNA molecules, ensuring that each daughter cell inherits a complete set of genetic instructions. This process, known as DNA replication, is fundamental to life, ensuring the continuity of genetic information from one generation of cells to the next, and ultimately, from parents to offspring. The discovery of the double helix had provided the key, and scientists were now poised to crack the code of life itself. The double helix model didn't just explain how DNA was copied, it also provided crucial insights into how this molecule carried the instructions for building and maintaining a living organism. Scientists had long suspected that genes, the units of heredity, were somehow encoded within DNA. But how? The answer lay in the sequence of those chemical bases, adenine, A, thymine, T, guanine, G, and cytosine, C, that formed the rungs of the DNA ladder. Imagine the 4 bases as letters in a genetic alphabet. Just as the 26 letters in the English alphabet can be arranged to create countless words and sentences, the 4 bases of DNA in their specific sequence form the instructions for building proteins, the workhorses of the cell. This flow of genetic information, from DNA to RNA to protein, is known as the central dogma of molecular biology. The discovery of the double helix provided the foundation for understanding this fundamental process, revealing how the genetic code, written in the language of DNA, is translated into the language of proteins, which carry out a vast array of functions within the cell. With this knowledge, scientists could begin to unravel the complexities of gene expression, exploring how genes are turned on and off, how they interact with each other, and how they ultimately shape the traits of a living organism. The discovery of the double helix structure of DNA in 1953 wasn't just a single breakthrough, it was a seismic event that sent shockwaves throughout the scientific world. It marked the dawn of a new era in biology, one in which the very essence of life, the mechanisms of heredity, were no longer shrouded in mystery. The double helix provided a unifying framework for understanding the diversity of life. Scientists now had the tools to delve into the genetic code and to develop new technologies with the potential to transform medicine and agriculture. The field of molecular biology exploded, fueled by the discovery of the double helix. New techniques were developed to isolate, analyze, and manipulate DNA. Scientists could now study genes in detail, paving the way for revolutionary advances in medicine, including gene therapy and genetic screening. In agriculture, scientists used genetic engineering to develop crops with improved yields. The discovery of the double helix wasn't just a scientific triumph, it was a watershed moment in human history, fundamentally changing our understanding of life itself. The double helix structure of DNA, revealed to the world in 1953, stands as one of the most profound discoveries in the history of science. Like the splitting of the atom, or the unveiling of the cosmos, it fundamentally shifted our understanding of our place in the universe. This simple yet elegant structure, two intertwined strands connected by a dance of chemical bases, holds the key to the continuity of life itself. The enduring power of the double helix lies not just in its elegance but also in its explanatory power. It unlocked the secrets of heredity, revealing how traits are passed down from one generation to the next. It provided the blueprint for understanding how life in all its diversity arises from a simple set of instructions encoded within the DNA molecule. The impact of this discovery continues to reverberate throughout science and society. It has fueled countless scientific breakthroughs, from mapping the human genome to developing life-saving therapies. It has sparked ethical debates and philosophical inquiries, forcing us to confront the implications of our newfound ability to read and manipulate the code of life. Yet for all that we have learned, the double helix continues to hold mysteries. It is a reminder that the pursuit of knowledge is an ongoing journey, one that leads us ever deeper into the wonders of the natural world. Consider this. Within every cell of your body, there exists a molecule so incredibly long that if you were to stretch it out, it would reach the sun and back over 600 times. This molecule, DNA, is not merely a blueprint, it is a vast library, containing within its elegant twists and turns a universe of information. The discovery of the double helix gave us the key to deciphering this information, to understanding the language in which the story of life is written. We have learned that the sequence of those chemical bases, A, T, G, and C, determines everything from the color of our eyes to our susceptibility to certain diseases. This knowledge has been transformative. In medicine, we are moving towards an era of personalized medicine, where treatments can be tailored to an individual's genetic makeup. In agriculture, we are developing crops that are more resistant to disease and drought, with the potential to feed a growing population. The ability to read and manipulate DNA has raised profound ethical questions about the limits of our knowledge and the responsibility that comes with it. As we venture further into this uncharted territory, we must proceed with caution, guided by a deep respect for the sanctity of life and the interconnectedness of all living things. The discovery of the double helix was not the end of a journey, but the beginning of a new one. It opened our eyes to the intricate molecular world that underpins all life, revealing a universe of complexity and beauty far beyond what we could have imagined. Today, the field of genetics is rapidly evolving, driven by technological advancements that allow us to study DNA with unprecedented precision and speed. We are uncovering the secrets of ancient genomes, tracing the evolutionary journey of life on Earth. We are developing new tools to edit genes with the potential to cure diseases and enhance human capabilities. The future of genetics holds both promise and peril. We have in our hands a powerful tool, one that could be used to alleviate suffering and enhance human potential. Yet this power must be wielded with wisdom and humility, always mindful of the ethical implications and potential consequences of our actions. The double helix, a symbol of life's intricate code, serves as a constant reminder of the interconnectedness of all living things. As we continue to explore the mysteries of the genome, Let us do so with a sense of wonder, responsibility, and a deep commitment to using this knowledge for the betterment of humanity and the planet we call home.